What's going on, all of my nursing brothers and sisters? I hope that y'all are having a wonderful day. Today we are going to start talking about something that's very important to all of us, and that is DNA and RNA on the ATI TEAS. If you haven't done so already, make sure you hit the subscribe button down below, and better yet, hit that bell notification. It lets you know when I post new content here on YouTube. And give this video a big thumbs up. That way other people that are looking for the ATI TEAS review videos know that this is a good video to help you pass it like a boss. Questions related to DNA and RNA cover topics including components of DNA and RNA, nitrogenous bases, and base pairs. You may also see questions concerning transcription and translation. Let's get started on understanding how DNA and RNA are important on the ATI TEAS. Every living organism has instructions for growth in the form of DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. RNA comes in many different forms: mRNA, such as messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. DNA, the larger molecule, stores genetic information for the organism as a whole. It contains the codes for creating new cells and is essential for the creation of new organisms during reproduction. RNA is much smaller. In fact, it is created from the nucleic acids in DNA. Its function is to help in the creation of proteins and amino acids, and it is found in ribosomes. It also acts as a messenger carrying genetic information around a cell and beyond. Chromosomes are strands of DNA and related proteins that reside in the nuclei of living cells. They carry the genetic information needed to create new cells and organisms. Amino acids are the building blocks of organic material. They are produced by RNA as the building materials for proteins, which in turn are the content of cells and cellular organs. Proteins make up enzymes which carry out work of cellular life like metabolic functions. Proteins also make up polymerases, which transcribe and transfer genetic material. Complex compounds are present in all organic cells. Nucleic acids are the core units of life. Both DNA and RNA are nucleic acids. A nucleic acid is composed of nucleotides. Nucleotides are nucleosides together with a phosphate group. Nucleosides are sugars, either ribose or deoxyribose, combined with either a purine or a pyrimidine. The 5 nitrogenous bases, adenine, A, guanine, G, cytosine, C, thymine, T, and uracil, U, are needed to make nucleotides. The sequence in which they appear allows genetic information to be stored in DNA and RNA. This information comes in 3 nucleotide groupings called codons. This is written as 3 letters. For example, CAG to show which nitrogenous bases they are composed of. With 4 nitrogenous bases, this leads to a possible 64 combinations of 3 letters in different orders. Purine bases, adenine and guanine, are bacyclic. Pyrimidine bases, cytosine and thymine, are monocyclic. Uracil is a form of thymine that replaces this pyrimidine in RNA. They form hydrogen bonds in a complementary fashion, meaning that purine always pairs with a pyrimidine. Purines and pyrimidines have different structures that allow them to form hydrogen bonds, crucial for the formation of DNA. One purine, guanine, bonds with a pyrimidine, cytosine. This is one of the base pairs discovered by Watson and Crick. The other is adenine and thymine. These base pairs are held together by hydrogen bonds. Cytosine bonds with guanine using 3 hydrogen bonds, while adenine and thymine require only 2 hydrogen bonds. These bonds connect the double helix of DNA. These codons contain instructions for building amino acids necessary for building organic structures. In order to remember which purine and pyrimidine connect together, I remember it like this. A and T are connected. Think of your at-home unit. You can also replace T in RNA. C and G are connected. Think of CGI. There are many minor differences between DNA and RNA. The major difference is their function. DNA stores and transmits genetic information for the use on the cellular or organism level. RNA transcribes and translates the genetic information into physical structures. DNA relies on 4 nitrogenous bases: adenine, guanine, cytosine, and thymine, whereas RNA, the T in thymine, is replaced by uracil. DNA has 2 strands of nucleic acids referred to as the double helix due to its physical shape. This doubling of genetic information plays a crucial role in genetic diversity during reproduction. RNA has only a single strand. DNA comes in many different forms. Mitochondrial DNA, for example, is only inherited from the mother. RNA also has different forms such as, like we said earlier, mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. DNA contains all the genetic information necessary for creating living cells and organisms. Transcription is the process through which genetic information is copied to make RNA. RNA polymerase, an enzyme created from a strand of RNA, binds with a DNA sequence during the initiation phase. This binding loosens the hydrogen bonds holding the double helix together. The bond section elongates with an addition of nucleotides. Proteins, called transcription factors, provide the needed material. The process terminates with a genetic duplicate strand of mRNA being released into the cell. Lastly, we have translation. At some point, genes must do their work. They contain the instructions for building amino acids, proteins, and larger cellular bodies. Translation is a process through which the information is put into reality. Translation means making a protein. The first step in translation is transcription. After the cell's genetic code is transcribed to an mRNA molecule, the information within the mRNA molecule is unlocked in the ribosomes. Ribosomes are situated in the cytoplasm of a cell or in the endoplasmic reticulum and are the cell's factories for producing proteins. The mRNA carries the 3 base pairs codons that dictate the type of amino acid needed for a particular protein. A strand of mRNA pairs with a strand of tRNA, carrying complementary codons during translation. The 3-phase process is initiation, elongation, and termination mirrors transcription, but it produces an amino acid. I hope that this video has helped for you to pass your ATI TEAS like a boss the first time. If you haven't done so already, I want to invite you over to my website at www.nursecheung.com. There's additional resources for you to help you pass this exam. We've got practice questions as well as PDFs of the PowerPoints that I show in these videos. If you haven't done so, make sure that you follow me on my social media. I am on Facebook and Instagram. And until next time, I hope that y'all are having a wonderful day, and I will see you all again soon. Bye!